This video is going to be about structures in cells that are involved in cell boundaries. An incredibly prominent cell boundary is going to be a cell wall. It sounds exactly like what it is. It's a wall for cells. It will support, shape, and protect the cell. Cell walls exist in prokaryotes, plants, and fungi. Now, these are not organelles. They're just merely cell structures, okay? So they exist in prokaryotes, they exist in plants, they exist in fungi. In plants, cell walls are made of cellulose, which we discussed as a carbohydrate. In fungi, they're made of chitin, which we also discussed as a carbohydrate. Cell walls do not exist in animals, okay? So that's another example of something that do not exist in animal cells. Cell walls do have pores. Usually think of a wall like you do for your house. It's not exactly going to be the same for cells. They do have pores. Water and nutrients need to get into and out of the cell. The cell needs to be able to remove waste and bring in things that it wants. So the cell wall does allow for things to move through it at certain locations, sort of like you have windows and doors in your walls so you can move in and out of different rooms. Cell walls also, of course, provide strength to plants to stand up against gravity. Remember, plants don't have bones, so these walls for cells really do help it give it structure. Cell membranes are another type of cell boundary, and you should remember, of course, from our very beginning here, all cells must have a cell membrane. So plants have a membrane and a wall, whereas animals only have a membrane. They will always be made of the same thing. They're always made of a phospholipid bilayer. So we're going to talk again about lipids in terms of a phospholipid and bilayer, meaning two layers. Okay, think bi in terms of bicycle or bifocals or something like that. And cell membranes, of course, regulate what comes in and out of your cell. It not only protects your cell from the environment and identifies, okay, this is the inside of my cell versus the outside of my cell, but it also lets things in or out, regulates what comes in or out to make sure that we get only things that we want coming in and only things that we want to leave leaving the cell. Let's talk a little bit about phospholipids. That beginning phospho prefix relates to a phosphate group, which is a phosphorus and several oxygen. A phosphate group is also negatively charged. And so we have what we call a the head of the phospholipid, and it is hydrophilic. And then the lipid portion of that basically reminds us of lipids. We have these fatty acid tails, and so these two sort of long chains, and we know they're going to mostly be made of carbon and hydrogens. And we know that lipids are nonpolar and that they're also hydrophobic. So we have these very interesting molecules. We have a hydrophilic head and a hydrophobic tail. And these come together to make a phospholipid bilayer, two layers of phospholipids. The hydrophilic heads point outwards towards either the inside of the cell or the outside environment. So here we have, let's just call this the extracellular space. So that's sort of outside, and then we'll call this the intercellular space. Okay, so we have outside the environment, that extra, like extraneous, and the inside the cell environment. Okay, and so those we can see these hydrophilic heads point towards the outsides. Here we have hydrophilic heads on one outside and hydrophilic heads on the other outside. So they point towards the environment, which is water-based. The cytosol is water-based and the extracellular space is water-based. Then on the inside, we have our hydrophobic tails, which point towards each other and the inside of the membrane. So we end up having this space that is hydrophobic that basically creates this barrier between the outside and the inside, not letting things to pass. And so this is really an amazing thing that creates our cell membranes and the cell membranes of all organisms. And so this phospholipid bilayer, as shown in this image, creates this cell that will allow us to separate the inside of our cell from the outside of our cell. This is a basic thing with cells. A cell is a unit contained within itself, and in order to have that unit, we need to identify what is inside the cell and what is not a part of the cell. And we do that with this phospholipid bilayer cell membrane. Now, of course, things want to be able to get through the membrane, right? We want to be able to bring food or other nutrients in, and we want to be able to bring wastes out or to secrete any other substances that we might want to. So, if a substance is able to pass through or move across the membrane, we say that the membrane is considered permeable for that substance. It is allowed to go through. If a substance can't pass, we can say it is impermeable, not able to go through. And so most membranes will be what we call selectively permeable. This is a big deal. It is selectively permeable. Some things can get in and some things cannot. We also say the word semi-permeable to mean the same thing. And this is really good. We want certain things to be able to get in, and we don't want it to have to put up a big fight for that to get in. But at the same time, we also don't want to just let anything into our cells, or anything out of our cells for that matter. So we want to regulate what goes in and what goes out, and so our membranes are selectively permeable or semi-permeable. And also we can talk about what we call the fluid mosaic model of membranes. The membrane is sort of like a fluid and a mosaic together. Stuff can move around within the membrane, which means it is fluid. For example, if I have a cell and I have something in the membrane over here and I want it to move to this side of the cell, it can move through the membrane to be over here. That's fine. So it's fluid. Stuff can move around within the membrane. At the same time, there are all of these molecules in the membrane, carbohydrate molecules, proteins, lipids that exist in the membrane that sort of dot this membrane, making it sort of look like a mosaic. So it's not only phospholipids, it's a whole bunch of other stuff. And so these items can attach to the cytoskeleton, interact with other cells, for cell-to-cell recognition, allowing cells to sort of interact with others. When we talk about membrane transport, it's going to allow things into and out of the cell. It's really a big deal. Let me see if we have a picture of this. Yeah, so this picture here sort of shows this fluid mosaic model. This item, which is a protein channel, can move over here if it wants to, but there's all this different stuff. It is fluid and a mosaic.